Let's go ahead and get started. Welcome to Data of the Dead. This is a fascinating symposium. My name is Jody Martin. I'm the Associate Vice President for Research here at the Natural History Museum. And it's a real pleasure to welcome some very distinguished guests and to welcome all of you. This symposium will highlight some of the lessons learned in the NSF-funded Thematic Collections Network DigIn grant, specifically the 2020 funded DigIn project to digitize marine specimens, which is wrapping up this year. Over the past 6 years of the project, new technologies and data sharing capabilities have emerged. We've learned lessons and times have changed. Now that we have, at a minimum, digitally cataloged marine specimens, we will explore how the worldwide community is building forward with data mobilization and extended specimen data science. We'll explore how new technology and data sharing promise to speed digitization and give digital specimens even greater value. We'll begin with short presentations by each of the panelists, and I'll introduce them in just a moment, followed by a panel discussion. After that, we're going to open the floor to questions from audience members here and those watching remotely. For remote watchers, please put your questions in the Zoom chat, and we will relay them to the panel along with questions from the in-person audience. Following our discussion, we'll host a workshop on applying AI technology to digitizing specimens, led by Christine Johnson of the American Museum. That will take place right here in this room, so just stay here for that. That will be an in-person workshop, and at that point, we will wish farewell to our remote viewers. Our speakers today is a very distinguished group. Dr. Regina Wetzer is Curator and Director of the Marine Biodiversity Center here at the Natural History Museum of LA County. Dr. Wetzer was captivated by the bizarre forms and crazy life histories of invertebrates very early in her career. She had the luck to end up working in one of the world's greatest natural history museums. Her personal research passion is in the marine isopod crustaceans, roly-polies that live in the ocean. For the past 6 years, she's led the NSF-funded DigIn program to digitize marine specimens across the U.S. Dr. Christine Johnson is lead digitizer for the Division of Invertebrate Zoology at the American Museum of Natural History in New York, whose collections date back to the 1800s. Dr. Johnson develops and implements scalable digitization for the division's estimated 23 million specimen lots. She has developed workflows incorporating high-resolution robotic imaging with AI-assisted transcription to capture label and card data and transform it into structured digital data sets. Dr. Johnson brings with her a deep knowledge of collections care, data migration, and biodiversity informatics. She is particularly motivated by the urgency to capture and share legacy specimen data, particularly from collecting localities that have now changed or disappeared. And Chris, it's a real pleasure to welcome you to the Natural History Museum. Dr. Libby Ellwood was trained as an ecologist, first working with natural history collections during her graduate research on climate change impacts on plants and animals, and she has never looked back. Now, Dr. Ellwood is Director of Education and Outreach and Global Collaborations for iDigBio. iDigBio is the organizing group for multiple NSF-funded digitizing efforts. In that role, her focus is on strengthening data practices in digitization and data sharing and building sustainable communities of practice. Beyond iDigBio, Libby is a leader in the International Partners for the Digital Extended Specimen, the Biodiversity Collections Network, the Society for the Preservation of Natural History Collections, and the Global Biodiversity Information Facility, reinforcing the international partnerships that underpin biodiversity research and conservation worldwide. And Libby, I can't tell you how good it is to have you back with us. Welcome again to all of you, and I'll turn it over now to Dr. Wetzer.
Thank you, Jody, very much. And I have my throat louder and my water with me. My voice is a little bit off, but we'll get through this. And it is April 1st, and he has made us a creation and spin of what we're going to talk about today. So some of you were here and saw Dean's presentation a couple of weeks ago, or a month or two ago from the RNCD. We talked a little bit about how the digitization project here at the Natural History Museum worked. I'll do a small recap for some of our guests that are online that didn't hear that presentation, and then I'll move forward and hopefully connect the dots between the DigIn program that we did here was done by some of the other institutions, specifically at the Museum, and then Libby will take us further on. So the DigIn program, we are cataloging for the first time we are making digital resources from marine specimens, and this excludes fishes and the mollusks, but includes everything else. And this was one of our very, very last collections that were to be digitized, and it's because they're very slow growing and very expensive to do. And again, most of our material comes from shipboard types of collections. In the perfect world, it's like very simple. The jar on the left would have a single specimen in it, a beautifully handwritten label with all the data on it, and that would be that. The middle specimen is a sponge, and oftentimes these are shipboard collections. They just have a station number attached to a number. So they present a challenge to themselves. They'll be taxonomically filed, you know what kind of sponges, based on where it's located, and you just have the number. Most of the specimens are in our collections, marine specimens, are very small, fortunately, so we can have very many of them, and we're very good at concentrating them into small spaces. And you would have many lots in any container jar. So we were a total of 19 institutions. We were funded for 4 years, which is extended to the 5th and 6th year. We were the lead institution, and I'll tell you why in just a moment. But these are some of our partners, and they ranged all the way out from the Pacific, from the Bishop all the way to Florida, to Harvard and across the US. And this was a distribution of like what we had set out to digitize. The green parts, the little green highlighted portions for the abbreviation of the institutions on the bottom axis are the percent of the collections that are already digitized. And then for this grant, the target was the yellow component. And as you can see, NHLA is on the far left, and we only proposed to do about half. We had the smallest amount already digitized of non-fishes, non-mollusks, but also have the very largest collection. So our marine invertebrate collections are only second to the Smithsonian in terms of its holdings. So, as I mentioned, a lot of our material is shipboard collections, and we focused on, because that is the majority of the collections, and that these resources are expeditionary based. So most of them are like that sponge specimen I showed you, which were very abbreviated field numbers basically in a jar. So by capturing the different kinds of resources and assimilating them, that is the expeditionary nature of this project. Again, over time, we've accumulated quite a few of them that we could identify, but this is also going to help us decipher our specimens. So in designing a digitization, because there's been many TCNs that preceded ours, and they were all very, very generous in giving us examples of like how they had solved their problems. But when it got to the invertebrates, we realized that we have very large institutions like ours, was quite a number of staff that are working on such a project, to small institutions that may only have had a half-time person or a part-time person, and that there were all kinds of different workflows and things needed. And I guess I put the slide up as an hindsight of the lessons learned. And the goal was to unify and be able to exploit all the workflows and make them all the same. And we realized we can collaborate, we can learn from each other, but the workflows were really very specific and also change over time. So I guess I encourage folks to think about, no matter what you're trying to do to capture data, you need to figure out what your resources are, what your beginning is, what your goal is, and when you think it's going to be complete, such that the nugget that you produce a larger picture as it moves through the data pipeline. So packaging it up in the end is a very important part. And for us, time turned out to be our biggest problem. We have access to many wonderful undergraduates, but then my undergraduates with somebody like this. I asked them to be the door monitors and now we're locked out. Thank you. Is that we needed to make things be able to move as quickly as possible. So we said we would do 320,000 lots, but we needed to make shortcuts of like, it was a minimal amount of data that we could capture and the biggest amount of time to get us the biggest benefit to get to that 320,000 component. And again, I turned to Adam, who made this very real in our first tests of what the speed of this whole thing would be, that if we just added collector names in this initial data capture, that would add 20 seconds for handling of every single specimen. And doing so would add 40 hours a week, an entire year's worth of one person. So that was the reality that we are, again, faced with. And again, some of our wonderful work study students that have been working on these things can tell you about how that goes. So the kind of skeletal data we chose to collect is the Darwin core data, very basic occurrence, event date, locality. Depth, of course, is very important for our kind of data, latitude and longitude on our samples, and then a taxonomic name. And again, this is all in Darwin core standards. So we have this skeletal data collection mindset. We needed to avoid moving through specimen jars. Moving specimen jars was going to cost us a whole lot of time and didn't have staging places to do that. Removing labels was also going to be time consuming. Needed to capture this data on a single pass, and the only way we're going to revisit a specimen was through the label photos that we had to take, and hopefully not ever having to go back to the jars, because that's just a whole other level of time. So what we did is we designed and created these workstations, these mobile workstations that we could put into our compact shelving. And here's one of our digitizers working away. There's a screen with the data acquisition data just to capture those fields that I had mentioned, the minimal collecting information that we needed. And the whole thing is optimized for speed and accuracy, and also making sure that the specimens are returned exactly to the shelf and the space and everything is put back together without damaging any of it. So how did we do? We got funded in November of 2020. You can see there's not much going on until about the second half of 21 because of COVID. And now we've had 2 extensions, but we're slowly making up our speed. The data that we collect, if we just collect the data, in other words, station data and taxonomic data, we can do that in about 35 seconds per specimen. If we have to take a photograph of the specimen label, that's 107 seconds per specimen. So it becomes quite variable. So where does this data go? It's the import of biodiversity data goes to multiple resources. The data has uniform persistence criteria, and again, you already, as many of you know, it goes to GBIF, Opus, Invertebrates, which is a symbiotic-based system, again, specifically for invertebrates, and iDigBio and Libby, definitely in that group. I'll give you an example of like one of these collections of what that looks like, and this is one of a collection in San Diego Harbor for the most part. It's called Navy San Diego 2021, for which the Navy collected material. We accessioned it here. We also were asked to generate digital, not only digital images of it, but genetic data. So there on the map are the localities, there's some pictures of some of our specimens. If you dive in, again, since this is from pure pilings and inner harbor stuff, an awful lot of polychaete worms, and there you can see on the GBIF download of what the proportion per family is and what the distribution is. But then putting in a context for all of the families of polychaete worms, and you see everybody's contributions, so you see them spread across. And yeah, there are those that are on land, and clearly issues with some of that alleys, and it needs to be clean. Obviously, there's the physical specimen itself, and then the specimen media, if there are images and so forth, and then the digital data that we just created, so that's like the first primary element of it. From that, you would obviously have field notes and field images, and if you were doing morphometric or isotope kinds of things. So, and of course, genetic sequences, and that's some of the things that I'm interested in. And then if you finally, have more of this digital capacity, finally past actually documenting that this thing is on the shelf, and we now know that, then you can get to this tertiary realm of like, okay, what can you do with it? Well, new species descriptions, phylogenies, phylogenetic data, behavior data, and all of those kinds of things. So the extended specimen, again, is a specialty of my colleague, Libby. So finding things physically. So because these things have not been digitally captured, the only way to file things is doing it taxonomically, which works great. But when you work with invertebrates and all kinds of phyla, which there's 32 phyla and only one of them contains the chordates, right? The diversity is very great, the expertise is an issue. We don't have that many specialists here in the museum, so we do the best we can. And with all the wonderful work of now doing complete phylogenetic analysis, I can give you an example of particularly marine crabs that have gotten a lot of attention. Reshowing and reorganizing them is just physically no longer possible. And also when things are super, super small, that's not necessarily useful. So now, rather than thinking about just databasing taxonomically, we know where on the shelf something is. Everything has a barcode in the jar with the specimen. The shelves are barcoded so that we can pull that data out electronically. And early days, I showed you the jar with the specimen of the sponge that had just the 4 little digits on it. Well, that's all they could do because there was no printers, there's no electronic means. We went through an era of where we could print laser printed labels and we put all the data into the jars. Now, again, there's so much more data that can be acquired. It's in databases. So there are labels, of course, in jars, but the data gets augmented and refined and improved in databases. So real database indexing is required to find. And then we come to where the specimen is on the shelf, and there's this crab right in the center of the slide, and from it, there could be pieces that go, molecular extracts that go to freezers. Other things go for longer term storage, maybe to a cryo sample or for genomic use in the future. There could be parasites on it, or epiphytes, or other hosts, or other symbionts. And they might be in different places. Genetic data generally goes to GenBank and BOL. The digital data is going out to GBIF and so forth. So again, this is the realm of the conundrum when one is starting off in a career of trying to figure out, again, when one's going a research perspective to having also responsibilities in the entire collection that the institution has. And you want to have as many wonderful hands and students and graduate students and postdocs also working with that data, how all of that is linked short term and then in the longer term as it gets explained. So with that, I am going to turn this over to Chris, who will tell you how things have happened at the museum and some of the innovations that she will folks are doing.
So, well, I'm really glad to be here to share with you how the workflow that we've developed in the Division of Invertebrate Zoology at the American Museum of Natural History to accelerate specimen and label imaging, as well as label transcription and databasing. And AMNH, I have to move to my first slide. AIM and H was part of the DigIn grant, but we also had some other smaller grants we were doing. So what I'm talking about is not just digitizing marine invertebrates, but also some of the other collections that we have. Ultimately, these data and images are going to lead to that extended specimen, whereby not only is the physical specimen important, but its metadata and other characteristics that extend the specimen into other realms. And as Regina indicated, Libby's going to be talking about that a little bit more. For many of millions of specimens and labels, our data are essentially locked in the physical collection. And if you remember back to COVID, many of us were literally locked out of the collections, the museum collections. And anything that wasn't in digital form was completely inaccessible. Being locked out of the physical collection is only one of the many risks that we face in terms of losing access to specimens and their associated data. Specimens with critical data degrade, ink fades, and specimens may be lost on loan or fall prey to pests or other natural and unnatural disasters. Because our collections are vast and numerous, it's often easy for us to take them for granted. But these specimens are effectively irreplaceable. For most specimens, there is no going back to yesterday to collect what was there then, and what was there then may not be there today. This Rocky Mountain locust back in the 1800s was so abundant, it literally swarmed by the trillions and was knee deep when they swarmed. By 1903, the species was extinct. We also tend to think of our collection specimens as being important only for morphology or species descriptions or elucidating evolutionary relationships. Not that that's a bad thing, but it's far more than that. However, these specimens represent participants in ecosystems and allow us to recreate snapshots in time, which allows us to make predictions about and preparations for the future. The task of getting specimen data from a slip of paper that might be no bigger than 10 millimeters in size with a 4-point font into a database field is daunting. Manual transcription is slow and tedious, and so there's a never-ending quest by our community to find methods that streamline and accelerate this process. And our solution to this problem was to image specimens and their labels using this robotic imaging system with AI-assisted optical character recognition, or OCR, for high throughput transcription. We took this gigamacro magnified robotic imaging system, which is actually intended to create a single large image from stitched together smaller images, such as the face of the cicada here. And I repurposed it to take relatively quick, high-resolution images of individual specimens or vials and their labels on a grid system, and developed a relatively efficient workflow using the images to create new catalog records and populate many of the fields in our division database, which is EMU, which is what I believe you guys use here as well. So I'm going to share a very quick video that highlights the process, and obviously we have some time to talk about this afterwards, and then I'll share the efficacy of the workflow and how this work can be expanded. Okay, so this goes really fast. So the robotic arm is on a system that moves the camera on an X, Y, and Z axis. Each specimen is staged in a cell with its labels and receives a barcode that becomes the catalog number, image file name, and is the common identifier in the workflow. The first cell is used to calibrate the run and holds the scale bars and the institution logo, and the arm is programmed to minimize the time it takes to move from cell to cell. The number of images taken is a predetermined trade-off between the desired resolution and the processing speed. The images are stacked with Xyrene Stacker, and the final image is renamed with the barcode in the image. We then use Photoshop Actions and Automate Batch function to insert and place the objects from the calibrated cell into each image of the run, which is what's happening here. Then the images are uploaded into the EMU multimedia module, and a skeletal catalog record is created with the image attached. Images are picked up by transcribers and submitted to Google Gemini or ChatGPT with a customized prompt for verbatim transcription of the labels in the image. The prompt tells AI what the format should be and that the first column should contain the file name, which is again that catalog number. Lines on a label are separated by a single pipe bar. Labels in an image are separated with a double pipe bar, and we instruct the model to preserve all characters as they appear, including sex symbols and diacritical marks. The verbatim transcriptions are then copied. They're preceded by AI generated transcription and the date that the transcription was done. And then the return transcriptions are copied into that spreadsheet, which is then used to ingest the data into the database where it's processed further. So, move forward. Start again. Not moving forward then. Not sure why. And I got stuck. Oh, there we go. All right, thanks. So by replacing our one-by-one imaging with this robotic imaging system, the number of specimens imaged and database per day was increased by 3-fold or more. And by having these stacked images, we were able to improve the image quality by 30-fold. Because the imaging starts at the base of the platform, we have a relatively clean image of both the label and the specimens. Replacing the human transcription with the AI OCR transcription of the label resulted in a 30-fold increase in transcription rates. It took volunteers 315 hours to transcribe text from around 7,000 images, so they were able to produce about 22 transcriptions per hour. When the volunteers submitted the images to Gemini or ChatGPT with our specific prompt, they produced a little over 11,000 transcriptions in 19 hours, so about 586 transcriptions per hour. The accuracy of both human and AI transcriptions was high, with almost 97% being accurate. Of the remaining records that had errors, there was no difference between humans and AI transcriptions in the number of records that had between 1 and 20 errors. Of the records that had greater than 20 errors per transcription, AI had more errored records than humans. However, in both cases where you have these really large transcription or this really large number of errors, it really didn't have to do with a transcription error, but actually the person putting the wrong transcription with the wrong record. And that was both in volunteers transcribing from an image to a record, or just putting the AI transcription to the wrong record. The power of AI-assisted OCR and transcribing labels is really transformative. It's fast, it's quite accurate, and saves our transcribers from the various ailments of hand typing label data, bad backs or eye strains or hands. And then once these text data are in digital form, this really leaves us more time to further process the data. And much more can be done depending on the needs. This is the only incipient potential for AI for this kind of work. For images with consistent fields like this card catalog here, data can be easily parsed into separate fields. On the left is the actual transcription returned from ChatGPT, and from this you can then ask AI to parse the data into the respective fields. So we have like the AI transcription, the card catalog, department number, accession number, etc. And you might even notice here how well of a job that AI did in reading that script, that hand script, which some of our younger generation today don't know how to do it anymore. AI was also even pretty good at counting specimens on a slide and indicating whether these objects on a slide were either full bodies or segments on a body. So with this particular prompt, we asked for AI to give the number of specimens in an image, and it did. And even more exciting is using these large language models to help find geographic coordinates for specimen localities. If you provide information like species and collector names, as well as collecting dates, this helps the model find other collecting locality data online, which are ether, which narrows down the points within a relatively small range. And these data can also help generate polygons by looking at existing species distribution. So a lot of this is, it's not pulling it from, nothing, it's pulling from data that are already out there. So the potential of this really is amazing. Having said this, AI is not perfect, just as humans are not perfect, even though we like to think we are. These models do more than, these models do make errors, and it really is up to humans to correct those errors. But with corrections, the models do better. For example, in this image, that red female sex symbol there was actually, you probably can't see it, we can't really see it very well from here, but in that first label on the top left side, it says trigona, and there's this 10 is circled, and AI picked that up as a female sex symbol. It wasn't picked up right away that was an error, so that any other record that had a label like in that position with the trigona and the circle was picked up as a female's sex symbol. However, once we discovered the error, and you tell it, say, hey, no, this isn't a female sex symbol, it's actually a number encircled, AI will go and then pick it up and fix it and actually read an encircled 10. So anyway, that's this is, there's a lot more to talk about AI, and this can lead to a much deeper conversation about using these models, and hopefully we'll have some time during the Q&A session to talk more about, I'm sure, those pressing questions about AI. So thanks very much. And Libby.
Hello, everybody. Thanks so much for organizing this, guys, and for inviting me. It's wonderful to see some familiar and new faces. Today I'm going to be talking about, building up of some things that were presented here already, and then kind of taking a step back and talking about some of the big discussions and ideas that are taking place more community-wide. So a little from both sides here. So I'm from iDigBio. iDigBio is a National Science Foundation project that's been around for 15 years now, and our job is to create disseminate the resources for digitization, which include things like best practices, or how do you image a certain kind of specimen, or all those kinds of things. So we've been doing that 15 years, and we are broken up into 6 domains, which then have been focused on various aspects of that, everything from the digitization workflows and that information itself, to what do you do with those digital data for research or education, or other things, as well as building some cyber infrastructure and other kinds of tools to be able to work with this. So lots of work has happened over time, but iDigBio is coming to an end, and there's still a lot to do, and kind of what's next. So today I'm going to focus in 3 general areas. I'll kind of go through these quickly, and then as time allows, we could dive in deeper afterward, or of course, always feel free to reach out to me at any time in the future. So with iDigBio ending, iDigBio has tons of specimens in our database already. All those are in GBIF already. So if you've been using iDigBio as a portal, you can always find those data in GBIF. And GBIF is an international organization that represents diversity globally. So it's a great place to go for all these data, and they're busy doing all these kinds of things. And in the top right there, they have, you'll see hosted portals. Right now they have about 58 of them. And those are subsets of GBIF data that are organized around a particular thing. It could be a region, it could be a certain taxonomic group, it could be a scientific journal. And then when you go to that particular site, you will see that subset of GBIF data and be able to interact just with those data. So iDigBio is working on that. So in the future, you'll be able to find iDigBio, as well as some of those associated resources that you may be used to using related to digitization through this hosted portal. So it's not out there yet, but I'm going to plant the seed now to stay tuned for that in the future. If you go now, you'll see some other cool things though. You'll see hosted portals. I know lots of folks here use VertNet, so you might want to check out what's happening on the VertNet side of things. But here's kind of some images representing the breadth of hosted portals out there. In the meantime, and just in general, really, if you're interested more in data publishing and how do I do that, or who can I talk to learn more about this, I will point you to this GBIF North America site. Within that, that's sort of the bigger umbrella for some of the North American based groups that are working in this realm. And so you can dive in from here. So lots of resources as well as data. And they often put on webinars on topics relevant to the North American biodiversity data world. So one to bookmark. So that's kind of the digital data space right now. And then moving on to build off of what Regina and Chris mentioned relative to extending specimens. So in the US a number of years ago, we really wanted to put a name to the thing that so many of us have been doing for so long, which is to say that we have a specimen and we like to attach information to it, right? Be it genetic information or pollinators of a certain plant and put that data together, whatever it is. And so extending specimens kind of within our own homegrown databases. So while we're terming that the extended specimen in the US, in Europe, they were kind of on a similar thought process. They came up with the term digital specimen. And so then we all kind of came together and we're like, hey guys, we're all talking about the same thing. Let's call it a digital extended specimen. So the extended specimen concept, you have the physical specimen as the core kernel there. And in the European version, it's a digital identifier representing that physical specimen. So sort of a slightly nuanced version of things, or the slightly different version of things, and then all the extensions from there are the digital identifications of the image or the genetic code or whatever, right? So similar ways, not exactly the same, similar ways of talking about the same idea. So then we're like, all right, this digital extended specimen, we still have some representation of the specimen at the core, and then when you kind of go up the chain there, the goal is to be able to link all of these kinds of data in a cloud, just have it all out there in a way that enables researchers to use not just the specimen data, but all those associated data in a quick and efficient kind of way. That's this is the goal. We're not there yet. So a bunch of us though we're talking about this. We meet about it monthly to see, all right, what are you guys doing about this? What are you guys doing about this? How can we work together to collaboratively not only sustain the projects that are working on various parts of that individually, but then these conversations that work to bring it together, because we need GBIF on board if that's going to be the biodiversity data side of things, from collections, from naturalist observations and all those kinds of things. But then also we want the folks who are talking about environmental data. We want folks, we're curious about the temperature, the water temperature from where that specimen was collected on that particular day. So having those folks at the table. So trying to think about how we can extend the specimen as far and broad as we can. And so here's just a subset of some of the projects and groups that are thinking about this kind of stuff. Some of what I've been talking about has been kind of in the primary data section on the bottom left, or the aggregated data section in the bottom right. But that top right section of associated data is sort of the folks that we don't always talk with as much, or we don't have the closest linkages with them built into sort of our usual workflow. So we've been working in a more focused way to try to make sure that all the data from all the various corners of this map here are at the table. And so a group that worked to bring some of these things together was the Biofair Data Network, which was a project from the Biodiversity Collections Network. And we kind of brought folks from the world of ecology and environmental data and genetic and genomic data and One Health and federal agencies, which aren't always kind of part of these discussions, as well as biodiversity data. So, all right, what does it take to make these connections, both with the people in the communities as well as the data? And so we came up with this kind of roadmap that identified some of those main things that we need to work towards. And this is from a recent publication, this figures from a recent publication that you can find a link to there. I think we're going to share these slides after, so no need to fret if you didn't catch all of that. And then the last thing I'm going to touch on today is kind of thinking about, all right, we have kind of all these siloed or disparate types of things happening, working to bring that together, but wouldn't it be nice if at a national level we could be more organized in how we're approaching some of these things? So it's not just maybe, oh cool, you have an extended specimen thing happening on your own laptop there where you're, pulling all this together, or, you're talking with the environmental folks and that's nice, but we could think about this, scaling this up and out kind of at a national level. And so the idea of an action center or a center for biocollections, integration and transformation has been floated, and that would be to not only take advantage of those interlinkages of communities and data, but even to think about economies of scale when it comes to things like storing images. Like if you guys are paying X amount of money to store all of your images of specimens, and another huge institution is doing the same, and all across the country, all these museums doing this, we're all to come together. Well, that's, you know, not only is it a ton of data, but then kind of the rates of who's paying what for what go down drastically if you're a little bit more organized in that. Not necessarily that that's our goal with image storage at scale, but just as an example, kind of if you think about working together to maybe solve some either financial or logistical issues that might be beneficial for folks. So in 2019 and 2020, folks got together and identified this notion of a biological action center, which in 2022 then ended up in legislation in this Chips and Science Act. Thank you very much, Biden, the Action Center for Biological Collections, which kind of said, hey, let's work together to do all this stuff, not just the data in the communities, but education and training and how to supply policy on and on. So that kind of didn't happen, to put it bluntly, because you all know what happened, shortly after that. There was a little bit of change in administration and where we're going to focus our efforts and on and on. So that didn't really go anywhere. But the fact that it was, it's out there really gives us something strong to point to that, there was a moment in time, there are a lot of people still thinking about this. This is still important. So it's out there. So you might see some familiar faces here. This was just right over there. Apparently this was 3 years ago, almost. Yeah. Who knew? But we all came together. iDigBio organized a bunch of webinars and in-person workshops to be like, let's just imagine for a second that this action center could be a thing. What would that thing look like? What kinds of what's important for us to work on, and what would you like to see that look like? And so we got together and thought about that kind of stuff. And just as of last week, we pulled all those literally hundreds of pages of notes together and identified a bunch of stuff that you all identified as really important for us to do. And so this you can find in Bioscience now, and it's things that probably aren't going to blow your mind, like, whoa, I never thought we should focus on that. But it does kind of give us a plan to move forward, not just within our community, but then when we're at the table with these associated communities that are, this is where we're coming from, what are you guys working on? And then where are those overlaps and synergies, and where do we maybe need to target our efforts to be a little bit more proactive and productive together? So read more there in that very nice Bioscience article, and thank you all to those who contributed to it. So that's kind of the gist of my talk there. I would be remiss if I didn't mention this digital data conference happening in a couple months that iDigBio is organized. Registration opens today. So go there, register, submit an abstract to give a talk, all that kind of stuff is going to be great. Thank you very much.
Okay, so move on to a panel discussion, and what we're going to do is Libby, Chris, and I, and Jody will come sit up here. We'll turn off the mic here, right? Dealing with logistics, and then we'll have some questions. Everybody sit again. Anything out the way.
Well, I should let the group play first. Okay, now I'm a person. So this is for Regina. Now that we're 6 years into what you guys started with DigIn, what are some of the lessons that you learned, and what would you do differently if we were to start again today?
Well, if we had the technology that Chris described, we would be doing a lot more AI kinds of things. I think the lesson for us and the things I'm struggling with the last few months is that all the data that our students have been collecting actually gets processed, and trying to make those pieces come together. And that's just an ever evolving, trying to do the easiest things first that have the biggest impact, and then just grinding away at it.
Maybe not have a pandemic.
But I must say, the pandemic was a blessing in disguise, and again, that might be a lesson to others as well. Because it really forced us for the first year and a half to think about how we would organize ourselves, and we ended up using technologies of the Google World Sheets and all of those things, Slack, all of the meeting recordings, the database, the website, all of that was created because we agonized because we couldn't come and touch specimens, especially for our institutions where we didn't have a card catalog like some of our colleagues did. And that made us just really, really refine everything before we got our hands dirty and started flailing and then having to reroute and redo and reanalyze. So in a way, I don't want to relive it because it was so stressful. But I guess launching into such a project of any sort of complexity, because it's an engineering project. And again, I look to Adam, who helped us remind us a lot about how that was, it's not research, it's nothing novel. You're trying to do efficiency, accuracy, and no damage to specimens. So.
Any other questions?
So, Chris, I wanted to ask you how you went down the path of this AI. Are you a techie person? I don't know you that either.
So, no, I'm not techie at all. I think my life is just trying to make things as streamlined and efficient as possible. And in all honesty, I had tried using AI or tried using OCR with Abby Fine Reader a couple of years ago, and it didn't work particularly well. I actually had a volunteer who actually did some kind of OCR with some of our session records. But then it was another volunteer who said, hey, Chris, I think we should try this. And to be honest with you, when this person said this to me, I was like, oh, listen, I tried to have you find me, it didn't work very well. And she said, well, I think we should try to do this again. And so we did. And the results were truly astounding. I mean, it was very, very astounding. And like during the workshop, I can share, a little bit of the not really the process what we've done, but we have a starting point. And it's just a matter of you giving the rules and telling AI what you want it to do. A lot of people, and I'm going to talk about this in the workshop a little bit, a lot of people think that it's an open-ended question and that it's going to answer all your questions and do everything for you. That's not how it works. You have to be very structured and give it structured data and structured rules for it to come back and give you something that's really. And so I can do it. Anyone can do this. Any other questions? Yeah.
Did you teach them the handwriting styles of different collectors?
So we'll repeat the question.
So the question was, did we teach it handwriting styles and rules? Not really, but what happens is when you submit these images and then it comes back and the image is wrong, say there was one particular example, it was in handwriting and it was WYGOD period. And I can't remember what came back, but it was like completely wrong. And so I said, this is really good, but this here is not XYZD, it's WYGOD period, which is short for White Gazinski, who was a collector of these species. Never got it wrong again. I mean, it is fast and amazing. And people can't even learn that fast, and that's what makes this. So we didn't teach it the handwriting. I'm assuming it already kind of knew it because somebody else has done this, but we have done nothing of teaching it script or handwriting. It picks it up really shockingly well.
I guess this question is for Libby, really. I think I get the feeling that part of this is happening, most of this is happening at fairly high levels. And I'm concerned about, I'm not sure how to phrase this the right way. Some of the communities in the world that need biodiversity data the most may not be up to speed in how to access that. I'm curious as to what steps we're taking to make sure that there's sort of worldwide access, even in those parts of the world that don't have the technology and don't have AI in their back pocket to do this sort of thing?
Yeah, that's a great question. So when it comes to things like, because there are a few different steps in the process, right? So if you think about data publishing and even getting those data out there, GBIF does a pretty good job of reaching out to different countries and helping them to be partners and to get local buy-in, which hopefully then trickles down to the later steps of the process to help those folks then use the data and apply it and know how to use it and apply it and all of those kinds of things. So there's that kind of side of things, but even just when you think about once those data are out there, you've got billions and billions of data points out there. How do you access, how do you even know to access it or what to do with it? And right now there is a pretty steep learning curve to be able to do that. But a project happening at the University of Florida is helping to improve that a little bit or to lower the learning curve through the use of AI and the chatbot called iDigBio. And in iDigBio, for example, you could ask it some basic questions to query GBIF as well as other databases like the Biodiversity Heritage Library, or if you're looking for publications or images or genetic data, you can ask it all those things and it will provide a more user-friendly version of those data that you can then work with. So you don't have to necessarily know what Darwin Core is all about and what those kind of enormous data files you're getting back from GBIF are all about. You can sort of cut to the chase a little bit more with a project like that or a tool like that.
I guess I'll also ask you to be in the data gleaning process, and we talked a little bit about that at the TAGWIC meetings in Bogota. The map that I showed is kind of disconcerting when there's no polychaete worms on land. So I'll let you address that.
Yeah, that's another tricky one. So when, because it's hard to kind of, there's so many steps in the process that even if you know that record is incorrect in one way or another, then you would have to then find out who provided that data point and contact them. And hopefully that's still the right contact person for that. And hopefully they remember how to, update that information, or they're the right person to update that information and to actually kind of go through all of those steps. And hopefully it wasn't, some kind of, well, hopefully somebody's still there to answer the phone essentially, and to do that workflow. There's no real way, like even if you are the world expert on that thing and you're 100% sure that that is wrong, and all you need to do is, let's say, change that latitude from a positive, from east to like a north to a south or whatever it is, then you can't do that. You have to go back to the data publisher and help them push that through in the right way. So it's not always easy, but it does. I guess that's, for better and for worse, that's part of the process, that these data are always evolving and improving, hopefully in the right direction. And I would also say though too, if you are a data publisher, to not let that stop you from putting your data out there, the fear of everything needing to be 110% correct and complete, but to put it out there the best as you possibly can and know that there's going to be times when you have to revisit those data and then have the systems in place to be able to do so.
It really is an iterative process. What we're doing is a very iterative process. I have a question actually for you as well regarding data going out there, and in the very early stages of this, we talked about the global and unique identifiers. Are those still being used? And how does anybody know what the value of those GUIDs have been over the years?
Yeah, they're definitely still being used, and I think we're also maybe getting to a point where it's more built in and baked into people's workflow. So it's not so much of like, please use an identifier, please make it, persistent and unique and all of these things. And, don't just make some random thing up that is not going to be unique, or that you want to change next week because you realize it doesn't work out. So I think we're kind of, as a community anyway, very much more advanced with identifiers, just even in the last number of years. And that's great, although, and I should say the resources now are also out there to be able to jumpstart your process, to be able to learn from the past. I think, what was the other part of your question?
have you seen the value of them sort of come back? So
Yeah.
we started with them and there was a large discussion about that when we started doing these like digitization projects. Has it been valuable to do this and is it getting
Yeah.
sort of cleaned up and narrowed down and?
Yeah, absolutely, right, thank you. So, and I think too, when we think about things like a digital extended specimen, that's also when it becomes even more important because our biodiversity data, our specimen having that unique identifier, then when it's, out there in the wild playing with environmental data and genetic data and all of this kind of stuff, it being out there for other folks to be able to find and use in their research, and to combine it with all these other data types that it just, as time goes on, it's becoming more and more important. And as time goes on too, it's becoming easier, I think, to create those identifiers and put them into our databases and then work with them within other biodiversity fields, but then elsewhere as well. Yeah, so definitely big improvements there.
Dean, anything on the chat box? I don't,
People are very quiet, but there are budgets.
I don't want to monopolize it. I guess this one is for Kristen. Is there any pushback from some of the curators at the American Museum? I can see if we could digitize everything tomorrow, that there are a lot of data scientists that would cheer, a lot of administrators would cheer and say we want. And yet a lot of the curators would say, yeah, but I know that 30% of those names are wrong. That seems like it's a battle still to be fought. It's a separate battle. At the same time, it's not one we should ignore. I'm just wondering if you're getting pushback.
Yeah, so I would say there are a number of people that feel that if the data isn't perfectly curated, perfectly identified, that we shouldn't put it out there. And I am of the complete opposite view, because I feel like science is all about building on what exists. And so if somebody has misidentified something, or if they're looking at cryptic species, if you don't put it out there, no one is ever going to discover it. And our collection there in invertebrate zoology, we have an estimated 23 million specimens. And there's stuff that unless somebody looks at it or even knows it's there, it's never gonna get looked at and no one's ever gonna know it's there. And even if it's unidentified, I mean, some of the stuff, even with these images, some people are like unhappy because the images aren't super, they're like, oh, we don't have the dorsal, ventral and lateral view, we don't have the antennal view or whatnot. But you can still tell from some of these images what genus this, the bee is or whatever it is. It is valuable and it allows researchers to come to look at those images and say, hey, this really, it doesn't belong to that genus, this does, I want to borrow these specimens. Instead of in the past, we would always, nobody knows everything. So as somebody asks for a loan and you don't know if all those species in that genus are really the right species. So you send the entire genus out with all the species, halfway around the world. Some will get destroyed, sometimes they come back, sometimes they don't. And this way, at least with an image, you can see that this is really what I want to borrow or what I don't want to borrow. It has value in so many ways. I hope I answered that question.
Yes,
Yeah.
thank you very much.
Sure.
And I guess I want to jump into your question as well, Jody, in that it's this iterative process. We've thought about it like, oh, we've inventoried it and then we're done. And that is by no means the case. And especially for what we did here, in that it was skeletal data that we took off of a jar and the expeditionary data. Some of them are very small geographic areas, and we chose not to try to decipher the exact second of the latitude and longitude that this location is in. And it may be the Ellwood atoll and it has an error radius, but at least you know that all this material was collected before we did the nuclear blasts or afterwards, and so that's where the value is. And if someone wants to go back and now knows that this stuff is there, it's easy enough to go and flesh it out and then repost it, and with a unique identifier, that data consistently rise with it.
Yeah.
Libby, I was wondering if you've seen many examples of institutions pushing out data kind of in an opposite direction, where instead of having someone collecting it and identifying it, but instead pushing it out as an Amelia but with a photo. And then seeing if people use that to identify the material instead of starting with the institution identifying it, pushing it out.
So yeah, let me see if I understand it and I'll rephrase it too. So you're curious about people publishing data with just a very high level type of identification and then leaving it open for people to come back to identify it. I mean, so this, you know, a little bit maybe like the iNaturalist kind of example where, you know, in animals or, you know, hopefully maybe it's like insect or bird or, you know, something, can then, help out and provide a finer scale, identification to it. But there's no similar method for collections information. And so the, most databases won't accept, that really high level type of information without something, a little more precise. Yeah, so I think that's sort of because that makes it really difficult to discover at all, just, you know, or it just makes things a little bit, more complicated down the line because it sort of flags it as, okay, this is definitely gonna need some work for anybody to be able to really use it. Because rarely, when you think about the downstream uses of these data, folks are generally more interested in things beyond just animals when they're searching for it. So you usually have to get a finer level of detail in there. So aside from like that iNaturalist model, which doesn't even become public until it has that finer level of information and it's become a research grade specimen and then it shows up on GDIF and things like that. So it's kind of like hidden from the world even at that high level. And so it's similar for collections data too, but that hidden version probably only exists on, you know, a laptop or maybe institutional database or something. So that's a good question, but I haven't really seen it play out in a forward-facing way like that for anything but like iNaturalist. Anybody knows of an example though, then
So I think what we'll do is take about a 10-minute bio break and maybe rearrange the chairs and then, you know, switch to the workshop. So this will be officially saying goodbye to everyone who's watching remotely, because I don't think the workshop is available on Zoom. And any other questions you've got, maybe hang on to those and we can talk about that during the workshop as we walk around. Is that okay with you, Regina?
sounds fun.
All right. Thanks very much, everybody. We'll reconvene.